Very good afternoon, all of you. I hope you had a good lunch. After lunch, listening to a lecture is hard anywhere. But I will make very simple and straightforward and not boring much. I hope you will enjoy my presentation. To avoid too much technical, you are all technical expert. I don't want to go into detail. If any burning questions are at the end of the presentation, we still have more interactive. So I come from, the CGR Center, Seat Alliance Biodiversity, where we look after the cross-cutting area of biofortification. I previously worked in millet and sorghum. Then later on, I look after all the cereals and legume, mostly the 7 crops of the CGR crop. So I'll be giving the overview on the biofortification, why we do or where we do, why should we deliver and deploy these cultivars through the mainstreaming approach. So biofortification came in 20 years back. It's 2003 almost. The Global Alliance of Harvest Plus established, and they made the big project on several crops. Majorly the food crops. The reason is behind is that it's a preventable malnutrition or undernutrition through the food system approach. So therefore, the focus only the stable food, not other foods. So you can see the 2 billion people are deficient in one or more other micronutrient. It's very serious, and if you take to the history, and these numbers is not changing much. So this is one or other, but we focus the 3 major nutrients. Why we focus those 3? And before telling that 3, why we need biofortification, some of the students and other people should ask before any of the trait to be prioritized in the breeding program. One is that we self-sufficient, at least in the production. And the historical achievement happened in 2022. More than 315 million metric ton food grain production. But if you see that the malnutrition or undernutrition, still we are not changing much. So people think that this should come from some other approach. But this is the approach. What we follow is that still we need to approach food system for the nutrition supply as well. So there are many layers of investment or the approach you can address the nutritional and health intervention. One of the largest and sustainable and cost-effective approach is the biofortification. So if you see the biofortification is that it's showing that at the bottom of the food pyramid. So if I ask someone what is the best strategy to address the nutrition or dietary diversity is the number 1, I would say. Because ensuring the dietary diversity everywhere in the rural vulnerable community is very tough in the current situation or even a decade ahead. Therefore, biofortification of stable foods makes sense, which reach the maximum population. So therefore, if you see that, this is the current scenario, the agriculture, stable foods contributes the energy and nutrition, almost 50% of the population. The rest has to be addressed by fortification, supplementation, or other health intervention. If you increase through these agriculture stable foods, you can reduce the investment in other, recurrent cost based strategy. So biofortification is nothing but increasing the micronutrient, essential micronutrient and vitamins in edible parts of the plants. So this is very important. In edible parts of the plant that can have the measurable health impact after regular consumption. So unless doesn't mean that we are making the biofortification. There are several approaches, but so far followed is the conventional. Is not against, the approach is against all the agronomic biofortification or transgenic or gene editing today. It's not that way. To easily reach the people, which is the best way, is the only way is through the traditional way of breeding. So therefore, they have chosen the conventional breeding so far, but there are investment and approaches already started beginning in other approach. But whatever I'm talking today, the cultivar development as well as the deployment all over the world in the entire global south is largely through conventional breeding. So coming to this important area of this training is talk about the climate or smart breeding. Is biofortification is not an exception. It has to also go through the stress and other traits of importance, which is part of the core breeding. If you're coming to the climate change, how does it affect the nutrition supply through the stable foods? If you see the iron and zinc and protein, nothing is left over. If you see the India is a more dense populated and also is the highly going to affect due to the climate change. So which means 1.7 billion people is going to be ordered by 2050. So which is at the level of if you do the carbon dioxide increase at 550 ppm. So around now we have 400 plus. So therefore, it is very critically look at the breeding program and ensure that nutrition should not decline further. Plus, CGAR and the other donor landscape looking after the nutrition in the crop development, that means crop improvement. So this is very important. It's recent, the COP28 and still going on. Is given that report very clearly that agri-food system. So which means agri-food system, all stable foods are the next frontiers of science and development, which need to align the climate actions. So therefore, CGAR, which is the, I would say, largest public funded network organization, not single organization. There are 14, of which 13 is being one CGAR now, is targeting 1 billion budget, for a year for all the crops, which has to be done. So therefore, the additional is come from the accelerated climate action now is 200 million. I hope many of you watching the news on the Bill Gates yesterday is announced foundation. So they have almost 800 million for climate action oriented research and development. So it is cannot be done by alone CGAR. It is need partner like you and your organization and your approach, everything. Therefore, it makes sense that we should work together, improving the productivity as well as the nutrition. As I mentioned, biofortification is not just another breeding program, it is a part of the breeding program. So which tells that, Harvest Plus works. It is a public, nutrition and public health by developing, promoting, just not developing, also promoting and delivering, scaling the biofortified variety across the globe, especially sub-Saharan Africa and Asia. And we do little presence in Latin America. So, but look that it's 20 years of journey. They have demonstrated this is a very cost effective and makes difference in the nutrition intervention, which comes, your following slides asked, at the inception of the biofortification, I said 20 years back. There are a lot of challenging. Is that breeding the stable food is can provide the required minerals and vitamins? If it does, whether it is bioavailable, the level, or at least what level the human being need per day, if that's met, then what happen? If farmers will adopt that variety, the rest of my presentation will give you the answer. So prioritization is very important. As I mentioned, that biofortification we cannot do every trait all at a time, but there is a prioritization. How does it looks? The biofortification priority index, which is analytical tool available in public domain. Anyone can choose almost 13 stable food crops. It is developed including 128 countries data, which means what this gives you. Say for example, here I put just select iron. It gives which country is the topmost priority for iron. To breed, to invest. It's not only for the breeding community and also for the donor to wisely invest. So similarly, you can choose another one. It is depends. The color intensity will tell how priority, how relevance, how would this match the production of the given crop and the deficiency among the population for the target nutrient and also the per capita consumption of the given crop. So we cannot deal the crop which is not being consumed at the region. So therefore, it's very critical. So it is easy to do, not by breeder alone. It has to be work with the nutritionist and various, socioeconomics, gender specialists, and population specialists, everyone. So in this initiative, nutritionist is the forefront and the decision making. The iron, zinc, provitamin A, all being entrusted by World Health Organization, which is the most relevant nutrient and vitamins to work for the human health improvement across the globe. Three nutrient prioritized, and these are all the crops. The dark one is the is the focus on the priority one, and this one is the priority two. So similarly in each nutrition, and what nutrient crops which center is lead is given for your information. Most of the CG crops is covered. Those are in food category. Continue to coming to the breeding program, and as breeder is always interested to have some metrics. How do we measure? So this is one of the important a slide which explained the zinc level of the zinc targeted crop, iron level in iron targeted crops, and provitamin crops. The light green one is that which is variation, genetic variation is available for that crop. It's not in the germplasm. It's the farmers grown varieties which is consumed, which is available in the market. But at a given time, this is taken almost 15 years back. So now we are in position to revise because the varieties pattern is changed now. So at the time it is given 16. For example, for rice, the target is 28. So already we demonstrated with the partners, various partners, 28. The dark one is met. This is the target. If you see almost everywhere, the target is met, which means there is a huge opportunity to mainstream this trait, the micronutrient. And as I mentioned again, nutrient target is simply not as a percentage over the check, like yield or something like that. It has to be meet EAR, estimated average recommendation, which is nothing but RDA. People use RDA in a medical, but being in agriculture and doing with the through food approaches, we always use EAR, which is 50% of the healthy population, where medical deal with 95% of the healthy population. So that makes sense that you have to make an improvement after regular consumption of your variety that are bred for that trait. So therefore, it is a key measurement for the nutrition, the policymakers. So it helps two way. One is that you know the level where you should reach, and also somebody can measure your progress where we are on the breeding program. How does it possible? It is invisible trait. Most of the nutrition except provitamin A modify the intensity of the orange or yellow color. But whereas for zinc and iron, which need some device to help you to advance those lines which are rich in micronutrient. So these are all the crops which is currently focused largely, but we have in others. Vitamin also using NIRS, but here I'm just focusing those high throughput XRF. Many of you heard about the XRF, which is 15 years before. None of the people is used for plant industry or agriculture industry, which is used largely for mining, a cement industry to detect the mineral composition of the soil or rock or the other particles. With the help of Oxford instrument and also the Flinders University in Australia, previously Adelaide University, they helped to devise that machine can detect plant sample also. So we have first generation, second generation, third generation XRF now, which handle 10 sample and 20 sample and 25 sample at a time. We can do the minimum number of elements and maximum number of elements we can increase. So therefore, it is critical to accelerate the breeding pipeline and selection for those invisible zinc and iron in your breeding pipeline, which is placed with the investment from the Harvest Plus Alliance. Almost 30 XR are placed all over the world, in addition to NIRS and HPLC, which can be used largely for provitamin A. But India is a country which has a maximum XRF. So because we have the diverse crop and every crop is facilitated in that. So after doing that, I'm sorry for the font size. It is, it's the targeted breeding approach. It's the 15 years of work is entirely targeted breeding. I define what is targeted breeding. It is not only in the mainstream. We have to demonstrate the selection for a given trait with all other demand-driven traits, either including market or the production traits. So therefore, one has to demonstrate. The demonstration has been done through this approach. Therefore, we call it as a targeted breeding. So this is very important, It's very critical to align the discovery phase and development phase and delivery phase. So the color can differentiate. But if you see the entire crop improvement, it's many actors and many disciplines play key role, including the priority setting and nutritionist. And there are, including, as I said, there are many investment going for discovery at the transgenic approach and other approaches, but conventional approaches took forward to make farmer or consumer acceptable once the variety is available in the field as well as in the market. On the right side, I indicated that although it is targeted breeding, it doesn't ignore that productivity traits. You cannot release any country variety trait alone. You have to meet other, the criteria of the yield or disease resistant or whatever is so called here in the maturity group, etc. If you see, go through the website, it is clearly indicated what is farmer preferred traits in addition to the nutritional traits. So therefore, we also given the how much nutritional benefit. So therefore, one should understand how value the levels and targets of the micronutrient in a given variety. So one should not confuse the public. So this is bio-fortified, that's not bio-fortified. Therefore, so it takes a longer time, but the investment made sense that several variety has been released. As a crop improvement expert or the plant science expert, you are all interested in the trait genetics. I've just given an example here, pearl millet, but this is applied to all the maximum cereals as legumes. The micronutrient, you all know that is the polygenic trait, which is quantitative measurement. So therefore, it is very important to include in the breeding program, not in the qualitative class. Being largely in most of the crop is additive genetic control. If it is some interaction, that is mostly additive gene interaction. So which gives the clear, allelic or selection based approach, one can improve the favorable alleles. And very interestingly, like or unlike yield, highly heritable traits. Therefore, transferring from the parent to offspring or the progenies, which is very high probability and stability. When you talk stability, there are two different stability, the trait introgression as well as this expression. One is G by E, it's not just E. Genotype by environment component of this trait is highly stable, which can determine the level of the micronutrient or vitamins, which is we assessed that is much better. I would say is double the amount of the yield traits which we are currently using. The nutrients much more stable in terms of GY interaction. And coming to association also will help the breeders to select along with the yield and other productivity traits. Being if you use that random population, which is not being focused so far this trait, which is you will get no systematic correlations. But however, in largely those are associated but not always with this negative significance. So if it is there in some of the random breeding population, one can use the large population size. If someone asks what large population, then we should ask how strong your negative association. Therefore, we can link that how much large population you should use 250 plants or you should use 500 plants. It depends upon how the linkage you have. The very high genetic variation is available to explore nutrition traits because this is very high genetic variation found in the germplasm and pre-breeding material. Also, the QTLs are largely reported in many crops, and these three micronutrients, the phenotyping became more robust and rapid. SNP markers being identified, and it is used for largely for F2, where we can much of the phenotyping. The F2 can select it based on agronomy, and so that the marker can help to link micronutrient traits. So how we took the fast track approach, developing a variety or the hybrid to demonstrate within a decade to reach the farmer. This is the regular approach anyone can follow, but we took the seed, a light seed parent. Pearl millet is like corn, cross-pollinated crop, which has seed parent and restorer parent. So seed parent is nothing but female B line, restorer is R line, pollen parent. So we screen entire the pool, and they found that those which are in qualified with the levels. So that's what we say the fast track approach. Then we make this the cross within the allied parents, within the pool, so that you maintain this, the heterotic pool of the male capacity as well as female seed producibility and cytoplasm. So then we went to the up to the A line, B line development and designated R line. It goes back to the test crosses. Then we evaluate from initial to advanced hybrid trial on farm, and then it is reached this level. Is nothing but the regular. But what we did is that scoop the right candidate for making the crosses. And similarly, the OPV is the intra-population being cross-pollinated. You can also play with that, which can much faster than your hybrid approach. So therefore, we release the first hybrid, OPVs, Dhanashakti through this approach. So when put all the inbreds, put together, if you see the restorer and seed parent and population progeny, and the IPC, which comes from directly as a germplasm, is all classified, and it gives almost 10 cluster, which very diverse in the low group and high group. So therefore, it is very critical for the breeding community to work on the germplasm which are ready to use and take approach. And some of the hybrid breeding consideration is very critical, So this is one slide which can tell you this is the released varieties, how you can see the Fe and zinc density is vary from 30 to almost, except to one population is 72, I think, here 73, and the remaining all fall. If you take average, it is 42 ppm iron. Similarly, for much lower in zinc, this is the germplasm progeny which bred for the trait. You see the scale how differ. So this is what is now is the approach to use when one can tap the genetic variability for future pipeline development. And coming to the approach of hybrid breeding, we always use the GCA like the earlier breeding program used. It is very critical to notice that very simple and much better than yield. And the parent iron and zinc per se, that means the mean of a given parent, if you see the line or tester, male and female, and this GCA effect, highly correlated, which indicates that you select based on the mean value also helps, because this is the invisible trait. It helps improving your population parental levels. And coming to that, how it is predictable, one has to test those test crosses, the mid-parent value of those given target nutrient, and also the actual hybrid level. If you predict those things, you see highly correlated, which again tells that it's additive genetic control. If it is the dominance, you can't get this correlation because the mid-parent value is additive weightage. And this hybrid per se, which tells that the dominance, which you can achieve through mid-parent heterosis and not much on the better parent heterosis. So happen in each generation, and how can you advance, and how the trend is look in the both seed parent as well as the restorer parent. You see the checks has been used for female parent and male parent Dhanashakti, which is come from the population. So it is clearly indicated that most of the population selected from F3 to F7 is gradually improve the mean of the population, and also you can see minimum maximum. Similarly for restorer. So this is the simple selection using the XRF screening is always helpful, and it is we are not losing the population mean, or also we are improving these the overall the minimum level of the particular seed parent or restorer parent. When it comes to the association, as I mentioned in the previous slide, we have to see weightage, not just for yield and also with maturity, flowering. It does, is no systematic correlation, is also good message one can breed for all three traits together. But if you see that, the days to 50% flowering largely they look, around 46 to maximum 50. So one can select those population or the material which can be promoted. This is that actual data collected from I think 2 years back in the advanced hybrid trial. So which has a high level of iron and zinc as well as the optimum yield along with this. So on the F4, F5 progeny tested also for the drought environment in the Rajasthan, you can see the level of the micronutrient, and also we identify some of the stay green traits which also informed for the drought resistant parent use. And some of the partners hesitate to send seed for analysis in the segregating generation or a different stage of the breeding material, including the private sector. That's genuine reason. But we have tested the hypothesis. One can heat oven the seed and they can send us, or they can analyze if they have the facility nearby, which tells that the germination is 100% in the control, and zero germination you can found, but nothing affected the iron or zinc control versus. So therefore it is a good method one can use, and fearlessly you can send those sample for the laboratory analysis in any stage of the breeding program. So very important, many of you know that how the private sector works in the hybrid, why they are being success. And most of the public program does the same, but it's need more organized way. The value of individual, as I mentioned in the previous slide, is always GCA based advanced. Okay, is we do a lot of test crosses. So therefore it improve the parent building. Once you improve the parent population, I think you will experience that the hybrid performance. And we also do that hybrid SCA advancement by testing. You can see that this is a hybrid testing. So this is the inbred testing. The female group as seed parent, male group restorer parent. One has to do intercross in the is two target here. One is that checking the cytoplasm restoration and also maintenance in case of the seed parent. So maintainer gene is very important when you are working and keep on advancing your breeding population. The two heterotic pools should kept separate, and the improvement happen only within that heterotic pool. So therefore it is test cross 1 and test cross 2 you can identify using one tester or two tester based on your target location. And this will tell us that largely one SCA based if someone want to commercialize and test the all India coordinated trial, etc. So therefore the new inbreds come out of it, this end of the cycle will go to again the process. So therefore female and male pool are maintained and enhanced regular interval. So we will not just keep that. So every traits are improved. Now we are talking about the climate resilient. One has to build that trait into the existing parental population. So therefore, their approach also rank based of their SCA, the hybrid performance is very important. Again, the mean based. So therefore, it is judged always by the multi-location year trials. So which gives a real picture of your population potential apparent heterosis. So by doing so, we ask ourselves how much variety we as hybrid developed are being competent with the existing check. So this is the bio-fortified millet hybrids. It's a different trial. So this is the private sector. This is the green color. The red one is the public sector hybrids. So these are all the hybrid which exceeded the performance of the hybrid in terms of the grain yield. Okay. So in addition to the iron and zinc. So it gives that a lot of opportunity one can combine and simultaneously select and breed. It easily achievable. And so far, at developed till 2017. I think they are being updating this numbers. One can see that how much number of elite, seed parent and restorer parent and donor line is developed for this trait. Being hybrid, and there is another, commercial angle or contamination angle question comes in front of the breeding community. One is that how the cytoplasm differences affect the micronutrient content. As many of the cytoplasm is not being commercially used. They have some reason, but some are not having the restorer. So one has to develop the restorer. So far, pearl millet A1 is much exploited, but A4 is being exploited now. A5 largely exploited for forage production. So we use this isogenic line. When I say isogenic line, which has to be, if say for example, ICMA, 98222, the same line should have the cytoplasm of A1, A4, A5 in a different version. But nothing is changed, only the cytoplasm source is changed. If you see that we have developed almost 20 to 25 hybrids using the 4 parents, which has that common, cytoplasm, it shows that the level, is not different. So this is the 2 location data as part of one of the PhD students, which gives that is, the B line, which is the maintainer, which gives 52, and the other cytoplasm is just closer. Almost the same for both iron and zinc, which means there is no cytoplasm effect, which altered the micronutrient level in the grain. Fortification is being mainstreamed. Is not just only breeding for the trait, which cannot make use in the crop improvement program. So therefore, it is very good, very important to screen for the existing prevalent disease, major disease. So major disease, the downy mildew, other one is the blast. So this is the downy mildew slides, which is we have screened for almost all the pathotype exist in India. So almost 90 bio-fortified lines screened in the greenhouse, and then which goes to the field in the multi-location. You can see how many location, almost 15 location, and then one has been selected based on that, the national criteria, how many lines, resistant number of locations and particular line. These are all the line which shows the maximum resistant in a major locations. So this is very critical to use always the screening protocol. So also we see the dual or triple resistant. If you see that 42 line were resistant to all the 5 isolates. So which shows that 10%, less than 10% of the downy mildew incidence, which is the target of the national levels. So this is a very key resource for all the location which can be used for the breeding hybrids. Similarly, the blast also, I don't have the slide for that. We does for the blast screening also, both in the glasshouse as well as in the field. This is just the genomic, many people quote that pearl millet is, I think 5-6 years back, is the only crop which released, marker resistant selection, which is 67 improved. But there are many came now, my other crop as well. And these are all the varieties, source lines is improved using the QTL introgression. So now they are also working, our genomic colleagues working on this, a blast as well. And this is some of the marker which can combine, iron, zinc, as well as the drought related, except to this one, and they have different marker for that. So this is the panel which is available publicly. Anyone can use these markers, a screen. As being breeding program, and we work as a long-term program, is one has to see what's the founder they have. Is assessing their founding parents is very important for any traits. So we did for biofortification. If is that the founder parents in the last 15 years has been used elite background, which has, each parent, it tells that how many number of times used in crossing. So this is the founder parents in the elite background. And also the population, which tells that how many number of times used as a parent, either direct or indirect. Indirect means it has to be crossed in other way. So it shows that, the maximum weightage is from this coming from this population, which clearly indicate that one can decide when one has to diversify the parental pool to improve the trait performance. So it is not possible, to reach a field and farmers without partnership. So this is a product testing always, work with the national partners who work on pulverized. Just an example, similarly for other crop, rice, wheat and maize. And these are also being tested in various countries. So this is very important to have the a testing partner and who can give the real feedback how the product is giving in their field. And also we conduct every year annual review meeting to get the feedback as well as the report on every line and the hybrids. So by doing so all, and recently couple of years back the national program has come to conclusion that these are all the trade priorities so far. This is the regional zone priority in the millet. A1 zone, which is the drought zone. A zone is the middle between the better endowed and the drought, and this is the highly irrigated and better endowed environment. If you see the trade progress, it's all downy mildews, blast, and sometimes ergot and smut. So far in the history, biotic and abiotic, always yield and stover is high priority. But now iron and zinc become part of the core traits, since 2018. So any variety to be released in the national system, they should meet the minimum level of this trait as well. So we take, starting from the discovery development to the policy level. So therefore, the work has been, is important for the product development as well as, somebody should refer some policy. Without policy, we can't scale up. So this is some of the example I'm giving from other crops. I asked my colleagues from CIMMYT, and they shared me, this is the interesting, somebody is maybe interested in speed breeding or accelerated rapid generation advancement, which is happening in all the CG center, I hope in the North Center as well. This is, you see, the speed breeding is reduced from 6 crop cycle to, 6 year to cycle of the breeding program. So one can see the year 1, year 2, year 3, year 4 will go to the actual testing, and one can also see the how fast is growing that the chamber and inducing the the lighting facility and the radiation, and that gives the flowering how quick. So this can only advance. And so we can't evaluate, but one can use the molecular markers if they have for the trait, even here. So this tells that the rapid cycle is really working for the wheat, and there are other crops are being working, but mostly the long day crops. The short day crops is being working still under discovery phase. And coming to that iron and zinc, the yield gain as well as the zinc gain in the breeding pipeline has been assessed over a decade, 2010 to 2020. So which tells that the entire, wheat breeding led by, CIMMYT. So by Velu Govindan is my senior, he is the alumni of TNAU. So he ran that, this program, and then he explained that, is 0.7, almost 0.8% gain per year for yield. So if you start in terms of kg, you see more than 100 kg, which is very good improvement. And what does for this type of analysis for nutrition is 6 ppm per year is increasing. So therefore, it is very good to measure. One has this is the blood-based, not the mean-based testing. So one can use this approach how the genetic gain is being used. Another way is using phenotypic selection and genotypic selection. We always use that, like yield check, you also use that, nutrient check. The BOLA 100 is the highest zinc found so far, which is being used, and there is the yield bar. So they meet that criteria, then they will select. Similarly, if you see the phenotyping, genotyping, how it does work, the color will indicate this is the discard, which is not being selected largely. But if you selected only based on the phenotype, you see almost 66% of the entry, more than 1,000 entries selection. Okay, only genomic, you can see here. Combining phenotypic and genotyping, you can see this one. This is the most critical part of the selection and advancement. So one can easily advance and confident to have the trait in next generation. This is one of the publication came recently, which tells that is how the nutrition level, you can see the yield has been improved in the long way, the breeding program, whereas this very recently started, it's a 10-15 years program which reached that it is a per day per plant productivity and iron and zinc, which tells that is ready to mainstream. And coming to that, climate change is not threatening that the breeding program we have to improve quickly, but one has to be careful enough to protect the native nutrient levels of a given crop. If somebody calls that as millet always nutritious, no, it's never be once we experience this. I will show some of the slides which affect the nutrition as well. So it is very important to connect the human and environment and plant nutrition. And so far there are three approaches being followed. One is the open chamber, which is controlled by, so this is the model we used, and we studied pearl millet, iron and zinc, how it is affected when you increase in different level of CO2. This clearly indicates that, again, I'm sorry for the font size. I try to show that, how the different rates, but I hope you can see the trend in color, the bottom is decline. This one is that in comparison to the control is the 550, which we don't have currently. This is the 700, which we have several years after. But the one is compared, which is the current level of CO2. So therefore, it's not much changes, but largely the grain is affected, not much in the fodder. So therefore, it's very critical to know one has to look very important to measure this trait against the climate change. And how does it looks, for the correlation? If you see that, maximum, the correlation doesn't change drastically in case of millet, in all 410 ppm, 550 ppm, it has affected, but very negligible. But somewhere, if you see, go to the negative in case of fodder, but still that has 700 ppm, we have to take several decades to reach. But what it is indicates that still it has a value to breed for the traits in against these. And also, this is the one paper we published, and it tells that, even within the panicle, the heat, this is about CO2, this is about the heat, increasing the temperature. It varies, from different position of the grain, which affect the different temperature. But it is, fortunately, this doesn't affect, as I said, the heat treatment, which doesn't affect the micronutrient level. And this study also proves that the heat is not much affected the iron and zinc in pearl millet. But we are replicating for other crops to know that whether trends is same or not. By doing so, and this is very important that, bio-fortified crops to reach the maximum global south, is very important to have that number, at least metrics, to inform the public. The more than 400 plus, 400 plus varieties being released to over 40 countries, and it's starting from, from the beginning to this year 2023, which tells that these varieties are being released by the regular approaches, which as well. However, releasing is the one way, but the breeder has to also link the scaling, which is missing in most of the cultivars release, chain. So this is the ICR released different crops. Very few are included because we follow the standards of the global. But this is maybe the national standards they follow, but this is Very few are maybe duplicated, but majorly is not because of the standard issues. So if you see, why I mentioned the release is not just a job to increase the farmer adoption. What is happen is an early generation seed, that is a breeder seed or nucleus seed to breeder seed or foundation seed is very critical. So far, this much seed only produced as per the ICR data, National Seed Net data, which needs that we need to increase the seed production to reach the farmers unless we cannot realize that potential of the bio-fortified varieties, and we started scaling with the various agencies. On top of it, we need the policy, as I mentioned, not just discovery, and also we link the policy related to the crop improvement, which supports that the such product development, either testing or scaling or procurement in a large scale. About 20 countries, if you see in the map, 20 countries by 2022 are endorsed the biofortified aspect in the crop improvement or the national policy interventions, health program, etc. So in India, so you can see these are all the area, the Maharashtra states, several states we covered, majorly zinc rice, zinc wheat, and iron pearl millet. Now, and the demand come from also from the finger millet. One no need to breed, which is finger millet highly calcium rich. But however, as I mentioned, what farmers grows and consumers, so that is very critical. So we found that there is a gap. So therefore, identifying those varieties, not breed at this stage. Identifying those variety and promotion is a great key aspect. And similarly, high iron lentil also is being part of the project. So at the end of the definition, I said it has to make measurable health impact. So gives you now how the EAR, daily iron needs, daily zinc needs, daily provitamins. So even the biofortified community, research community doesn't realize in the inception, they don't achieve this much level. But they find, after several clinical studies, both conducted independently as well as dependently, it gives that iron crops 80% of the daily requirement. If you consume the per capita level, say for example 200 gram per day millet, the biofortified millet or biofortified beans, which gives the huge potential to scale. Where it comes to zinc, you see 50 up to 50 is giving 70%. So this is not just selling point, it makes the impact on the human health. Therefore, we justify the biofortification is that trait is doing its job. And vitamin A you can meet almost 100% because the bioavailability percentage is very high in compared to zinc and iron, which has interaction with the other elements, including phytate, other polyphenols. But whereas vitamin A is very high. If you ask that, what does is that much we need, but how it is improves, you can see iron biofortified crops. It reverts in the 3 experiment, the iron deficiency. And it is proven that cognin in this, the brain development, function is very high in work efficiency when compared to the control. Whereas zinc is also does that, your height of the age for the children and illness, it also related to diarrhea, fever, vomiting, etc. That improves. Similarly, vitamin E. This is all not done by the crop community. It is all done by the medical as the nutrition community, independently both in Asia as well as in Africa. So that gives the boost, that gives the confidence to mainstream these type of traits in the competitive varieties. So how Harvest Plus or biofortification align differs with the existing breeding program, either in the public and even including the private. It does not stop at the seed release. It ensures it reaches the retailer, either in the seed form or in the product form. So it is not the crop improvement community job, but there is a different division is present and different partner capacity we have built. One is the crop trait development, mostly the crop research institute public, I said again, public, they do a lot of research and they do certain crop development pipeline, then goes to private, and then they will take care of the seed multiplication and business, and the maximum effort will stop at this release. But this has to be strengthened, as I mentioned, is the number of variety release is the one metric to tell the breeder has we achieved this. But one has to achieve how much population or how much area is grown by a given variety. That tells that are we breeding the right product or right use. So therefore, the seed distributor and crop protection and post-harvest handling, because some of the processing also will improve. Some of the marketing traits also is attract the breeding, trait incorporation. Therefore, it is very important to have the feedback as well as the chain keep going till the consumer, as Harvest Plus does with the various more than 100 stakeholders partnership in the world. So main important that nutrition valued in crop improvement and seed system investment. So therefore breeder cannot be overloaded with a dozen traits, but that makes sense that whether it is value in the market or the consumer preference, it should do that. And this is okay, once you have done all the biofortification product is available, how we can classify in the market, on what basis we can buy the procurement program or intervention program. If you see the seed, we have several class or policy or standards in terms of research as well as the delivery. Okay, in the floor after processing, people will pack it and they have several standards, FSSAI, etc. The grain which farmer produce and goes to market, there is no standards, and there is a big valley and big gap. So we understood that, 5 years back, and Harvest Plus developed that pretty standard institution and Global Alliance for Nutrition Gain. Okay, is very much develop the standards one can use the procurement, which should not differ from the breeding standards. So therefore, we make sure that we are connected. So how it is possible? Zinc enriched wheat maize with manual specification, which is publicly available. One can browse it. Is for zinc separately, iron separately, and as well as the provitamin E separately. So each has a different number. One can look at it. It gives a very clear indicator how one can during the procurement in the market. It's very much important to have that, but we are clearly told that this is for enabling commercialization and scaling purpose, but that should not confuse with the breeding target. So this national, ICR also come up with this manual which gives that, some information of the released crop varieties. So that is more of, food system approach. As 20 years, as I mentioned, Harvest Plus initiated what has been impact so far. So as I mentioned in the snake diagram, it's reached the retailer and also done impact assessment. That how far we made a difference in the society. Is that this report indicates that, it's almost starting from 2000 now till 2023, we are reaching 100 million people. It's only at the on-farm, not the buyers who consume. So only the people who grow and consume. So therefore, our aim is to reach 1 billion by 2030. It is not our aim, I think our means everybody, including the partners and donors. We have to keep continuing this trade in progress and mainstreaming. How it is possible? Only through mainstreaming, not through targeted breeding. So therefore, it's very critical to measure the progress, both in terms of the breeding as well as in terms of reaching the farmers. How the mainstreaming we advocated and developed, how quickly the program can do, the product profile. They made 60 ppm minimum and 35 ppm minimum zinc in the parental advancement. Replace every time 10% of the parent every year. So therefore, your allied pool is increased for the trade, and use XRA, which is equally efficient and diagnostic method like your markers, diagnostic markers for early generation, especially up to or F3. So today we started from here, and we are here in the bio-fortified, but we need to merge through the mainstreaming population in terms of the breeding population and not the human population. We have to reach this. How it is done? So we hypothetically proposed that from now 10 years by 2030, we have to do full-fledged mainstreaming. So therefore, targeted breeding will disappear. So that's the plan. So therefore, funding will be diverted largely towards the mainstreaming and the local testing, etc. Is how does it possible? We have to move from the periphery area to core area of the breeder list. Those crop has been proven that can be improved along with the yield and disease resistant. There time for negotiation, it is very important to include. Therefore, CGR made promises that product profile, just blueprint of the breeders who can develop the variety or population which can be released after 5 years. Develop the blueprint now. So this is the approach they are following, and just like the private sector. So this also this is replicated in most of the public organization in major crops. Private sector involvement to make the capacity building for the NARS. So therefore, the investment is slightly, modified. The landscape of the investment from the donor, government has to be aligned through the policy to make sure that this can be improved or integrated as a core trait. So based on the discussion and various delegation, now we found that I think this, comprehensive unique case for CGR, as I mentioned in the beginning of the slide, 4 billion investment is our target 2030 per year. So to reach that, and what scale they use, for every $1 investment, either in the breeding program or any technology in the CGR, that should return worth $10. But biofortification already proven that, $1 investment gives $17 benefits. So therefore, they endorse the biofortification mainstreaming through product profile, which I mentioned earlier. So therefore, it is very important. These are the people who makes the very good contribution in the food security. Professor, Ejecta, and he is in a Purdue University professor. Now is a World Food Prize Selection Committee chairman, and she is the World Food Prize laureate. The climate change. Why I put this is the climate resilient. We are working with her some of the project. And he is the envoy, special envoy, Dr. Kerry, in the US government. So therefore, it is very critical. One has to think that both food and nutrition security align to the climate change area. Slowly, it's not just for funding and also for making useful for the network, facility and capacity. So I think I can conclude now. Very simple and straightforward messages. Biofortification is science for delivery. It's not stop at the science of discovery. So therefore, ensure this is not that deteriorate like, genetic erosion. It maintains. Once you put the genes, alleles, it maintained. As I mentioned, this one can carry forward. And it is the best cost effective. It targeted for the rural population, who is the really vulnerable people. Make sure that strategy is still sustainable for the entire supply chain. As I mentioned, this is a core trait in the product profile, being not compromised. If you compromise, then other words, the food system so far compromised in the released varieties, 30% decline in the modern varieties, both rice, wheat, in mega crops. In other words, they compromise the nutrition to build the yield. So therefore, it is very important to start now, at least stop that declining trend to make that part of the core traits. As I mentioned, the climate change is coming up. One has to be prepared, so not to be very, one has to be prepared for that, further disturbance happening in the mega staple food crops, which is dependent large of the poor marginal farmers and consumers. So therefore, what's the challenges? It is still challenges ahead. Need modeling and nutritional indicators, both for breeding as well as the climate vulnerability prediction. So therefore, it is very important to tell the people how the prediction is support decision making. And strong characterization of the gene bank and the pre-breeding material, which require additional funding to the breeding community or the nutrition community to continue the mainstreaming. And engage public and private partnership policy. As I mentioned, policy always give the people strength our support encourage that doing the nutrition smart varieties in future. Therefore, the central goal of the crop improvement and the food production has to be optimized for nutrition security as well. So therefore, to save the some of the public investment in the nutrition and health. So with that, so I thank that for the opportunity by the organizer CPPGTNAU. So I always remember these two phases who talks more on the food security, and they ensure at the bottom of the food security is the nutrition is still not addressed. If you go and refer any of their article, they always mention that the hangover of green revolution effect of the major crops. So therefore, one has to improve the nutrition through breeding, which is the sustainable way. Thank you. If you have any question, I'm happy to respond.